Right, good morning, good afternoon, good evening, ladies and gentlemen. Welcome to this webinar about the transforming cancer detection with liquid biopsy. My name's Steve Hawkins, I am your moderator, and I'm just going to, in a moment, introduce our speakers, and then what we'll do is we'll go through a set of questions to actually ask the speakers. What I will say is if you do have any questions as we go through this, anything stirs and you want to ask questions, please add them to the information. There's a chat box that you can actually fill those questions in on. And if they're germane to the actual conversation at the time, we'll ask them then. And if we get some time at the end, I might ask some of those questions at the end. So if not, we can always ask the panelists later on just to fill in some information for you. So first of all, let me introduce you to our panelists. Our first speaker is Chris Sale, and Chris Sale is from Nonacus. He is a co-founder of Nonacus, which started in 2015 with a principal focus of developing non-invasive DNA testing that would change the landscape of genomic diagnostics in the field of prenatal diagnostics and oncology. He has over 20 years experience in the biotech industry, including previous roles in Illumina and Agilent Technologies as a clinical diagnostic business manager and consulting genomics account manager. Secondly, we have Hongjun or Harry Yang, and Harry is, or he joined DiaCarta a few years ago and is the SVP of companion diagnostics, and his focus is on the next generation of companion diagnostics to facilitate anti-cancer drug discovery and development and the implication of precision medicine. Prior to DiaCarta, he was Executive Director of Personalized Healthcare at AstraZeneca and was involved in the launch of Tagrisso, Alaprobib, and Imflinzi, which are companion diagnostics. He is very well known or very well established and a passionate, dynamic leader in the biomedical and pharmaceutical industry and academia as well. And he has served as an executive on multiple startup companies and institutes in both China and the US. He was the original developer of electrochemiluminescence ECL technology, which I've actually used a lot of in my past, and its instrumentation, which was exclusively owned by Roche Diagnostics. He is a pioneer of flow-through DNA chip technology and an instigator of the global human genomic HapMap program, gene expression database, NGS platform design, and biomarkers and companion diagnostics for precision medicine. He is one of the Chinese scientists, or one of the first Chinese scientists to publish as a first author in a Nature Biotech paper, which dates back to 1994. He is an active member of various professional organizations and a co-founder and founding chairman of Precision Medicine Committee in the Chinese Society of Biotechnology, the CSBT. Thirdly, we have Shidong Jia, and he is the CEO of Predicine. Prior to founding Predicine in 2015, Shidong spearheaded the oncology biomarker strategy and drug and diagnostics co-development at Genentech. He previously held various senior research positions focusing on translation and clinical cancer research at Novartis Institute for Biomedical Research and Harvard University. Shidong was actually awarded the PART Investigatorship Award from the Dana-Farber Cancer Institute, and Shidong actually holds an MD and PhD from Harbin Medical University and Peking Union Medical College, and completed postdoctoral training at the Dana-Farber Cancer Institute at Harvard University. And then last but not least, we have Florent Chang-Pi-Hin, and he is a leader in the life science industry, working as a global research and development manager here at Meridian Life Science. So he has been working in this area for over 10 years now. In fact, probably over 14 years now, because he's a few years more advanced than I am with the company. And Florent's group is based in London in the UK, with a focus on developing new critical reagents such as qPCR and isothermal enzymes and mixes used for the global diagnostic market for the development of molecular diagnostic tests. Their recent market releases allow for room temperature stability and greater inhibitor tolerance for diagnostic assays, which are now playing a crucial role in the detection of SARS-CoV-2 worldwide. So welcome, panelists. So now what we're going to do is we're going to open up the questions. And the first question for you is, why is liquid biopsy considered a game changer in the field of cancer diagnostics and precision medicine? And what I'll do is I'll open this up to Chris first of all, just to give your interpretation of this and what you actually think.
Thank you, Steve. I think.
Oh, Florent, you go for it.
Yeah.
Sorry, I apologise.
No, it's okay. I was just wondering here, maybe it would be good to, because it's a very wide question. And I think there are a couple of notions maybe to define here. I think for me, the liquid biopsy needs to be defined. By liquid biopsy, I think we mean, of course, blood, but also all the other fluids of the body. Whether we're talking about CSF, we're talking about potentially also urine. These are all liquid biopsies. And then also, I think you were saying why liquid biopsy could be a game changer for the field of cancer diagnostics and precision medicine. But for me, it goes beyond cancer diagnostics, because if you think about cancer and the different form of tumors, you need to think about the early detections to detect whether or not there's a cancer, but also then the further application with the prognosis of the cancer, what type of cancer, then the selection of the treatments, which is feeding into the precision medicine, but also finally the monitoring of the efficiency of the treatment. So I think for me, liquid biopsy take into account all those elements. So I don't know, guys, Chris, Shidong, or Harry, if you want to share your views on that, but I think it's a very wide field of application.
Yeah, I mean, thanks, Florent. I mean, I'll add in a few points from my perspective of why it could be a game changer, and is a game changer. It is the liquid biopsy, if collected correctly and processed correctly, and then analysed with an appropriate technology, can represent a more accurate representation of the actual tumour than could be obtained by biopsies, by physical biopsies, where you may biopsy an organ or a tissue and get a misrepresentation of the entire tumour. Biopsy just may not be achievable or possible to achieve and obtain.
Yeah, it's a good point actually to consider this. Is the fluids, the body fluids, it goes everywhere and reach out every part of the body. So that heterogeneity of the tissue is a key consideration, I think, and the game changing is the key. Yeah, makes sense.
So does that mean that when you're looking at this, you're looking at specific fluids for specific cancers, or is it more you look for blood first of all and then you look in other fluids?
I think, Steve, it's fairly well published now that the different cancers shed the tumor DNA at different levels into the bloodstream. So blood may not always be the appropriate sample source for all cancer types. And also blood is not truly non-invasive. Of course, it's minimally invasive. And so there might be more appropriate and better sources of the liquid biopsy sample than the blood.
Right, right. And is a lot of processing needed for this? Because of course the worry is that you've got very, very small amounts. And if you're having to purify everything from these tissues, that you're going to first of all purify those small amounts away, and secondly you're going to change the ratios. If you're looking for different biomarkers, you're going to change those ratios, which may cause problems.
Yeah, Steve, I think that the game-changing element of liquid biopsy, of course, is the meaningful of the data you're going to have, the sampling itself. Yeah, you're going to sample something that capture the whole body states, not just that particular volume in the body, as Chris mentioned. But also it's the ease. You know, you don't have to go through a biopsy every week to monitor the efficiency of a drug or a treatment. So if you think about the comfort of the patients, that's one of the elements that makes me believe that it will change the game to some extent.
And I think, Florent, that point about the comfort of the patient flows through the entire patient journey, where as you mentioned, in the early cancer detection setting, a liquid biopsy could be used to triage the patients correctly, so that those patients that don't require an invasive oscopy, like a colonoscopy or a cystoscopy, can avoid those painful methods, right through to the post-chemotherapy, post-surgery, post-treatment monitoring, where, as you said, physical biopsying every 3 months is just not possible or feasible.
Indeed. Shidong, perhaps you'd like to say a few words.
All right. So I think the liquid biopsy is absolutely the gold standard in precision medicine. And clearly, as the earlier speaker mentioned, that its non-invasiveness and also its robustness in detecting cancer-related variants in circulation, such as blood or urine or other body fluid, that just opened up a brand new avenue and to detect the cancer, the root gene, and then also select the target therapy, not only for treatment selection, but also for therapy monitoring, and also the minimal residual disease, and towards early cancer detection. And liquid biopsy right now is a new poster child in personalized cancer care.
Yes, well said.
I was also thinking something, if you take the testing in its globality, you need to think about the context of that particular testing. And if you start using liquid biopsy for also other applications beyond the oncology markets or applications, and I'm thinking about STDs or infectious diseases, then it gets into the usages, and people are used to the processes, and then you just develop your technologies around that. So that's when something starts to be working, when something starts to be easier, then you can see the expansion throughout the application.
I guess that would actually help with the approvals as well. If you're using this technology in different areas, it's going to be easier to get approvals from the authorities to use it in other areas, because they become more comfortable that they are technologies that actually work as well. So that's going to be easier for you to get your assays through regulations. Okay, well, we'll move on to the second question. And the second question is, which technologies do you favour for liquid biopsy and why? And I will ask Chris actually this question first this time, because there's obviously there's different technologies out there, but for yourselves, which ones do you actually favour?
Yeah, thanks, Steve. So definitely at Nonacus, we take a pragmatic approach to which technology to use, really based on what the needs are. Which is the best method to diagnose or undertake the analysis of a sample it's different depending on, or could be different depending on the type of cancer. So not all cancers are alike. They have very different mutational spectrum. And therefore, the number of loci you need to interrogate or analyze within a patient sample could be very different between one cancer and another. And that may lead to a certain technology type being required. Equally, the differences between different technology types in terms of their ease to implement, the technical capacity of laboratories to implement them, and the cost burden or analysis and informatic burden between NGS and PCR, for example, again, are very different. So may suit different customers or different healthcare markets. So it's not an easy one to answer, but here at Nonacus, we very much look at answering the clinical question with the best technology type to answer that question. So we actually develop our PCR and NGS-based methods for analysis.
And I think that's a key element. Yeah, the diversity of the questions that needs to be, answered with different technologies. And we've seen that throughout the years, with different, whether you want to look for mutations or presence or absence, or deletion, you use different methods. And I think that that's not going to be different here for a liquid biopsy.
Yeah, I think certainly that, it's a trend moving forward. Like, the liquid biopsy right now, become the very useful complementary the technology in addition to the use of the tissue, and also the NGS and also the PCR test are the platform that to address the similar question depending on the need. And definitely there is the pros and cons and in the clinic. And then right now that with more the personalized the drug being approved by FDA, I think that the technology is being identified and to best approach the clinical questions, such as the PCR sometimes is more than enough for the single point mutation, but when you look at the multiple, and the NGS is a better approach, and to address the 10 variants or 10 different genes through the one test. And then again, when tissue is the issue, then liquid biopsy stand up and provide the real-time the molecular diagnosis. So I think that the right now precision medicine just come to the right time, and with the endorsement of the new technology, especially liquid biopsy.
Yeah.
And maybe we have simplified a little bit in the discussion the technologies. We always talk about PCR, but just with PCR and just with NGS, you need to look at DNA, RNA. You need to look methylations or any methylations. So even when we say those simple acronyms, there's far more technology beyond this. The variants are huge.
And a question for all of you. Obviously, we're talking about the molecular side here, but obviously there's the proteomic side, there's the metabolic side, there's the lots of different SNPs and things like that. Is it better just to concentrate on the molecular side, just the DNA, or do you think, and this will probably go to a question later on, so you don't have to answer this if you don't want to, but do you think that we are just blinkering ourselves at the moment, just using the DNA, or is that at the moment for the position the technology is in sufficient for what we're doing? And as the technology develops further, we're going to be bringing those other areas into it.
I think from my side, Steve, it depends on the area you're focusing on. So ultimately, I think we're all working in an area we want these cutting edge cancer tests to be adopted and available to as many people as possible around the globe. And to do this, of course, workflows need to be as simple as possible and costs need to be as low as possible. So you're absolutely right. You can interrogate RNA, epigenetics, proteomics. There are lots of different tests you can do. But ultimately, if one test and one methodology gives you enough sensitivity and specificity, then it's enough. I think the multi-omics approaches will be best played out in that early cancer detection setting in the kind of larger populations or slightly enriched populations where single omics approaches have not shown great enough results at stage 1, stage 2 cancers. I think in that MRD space, single omics approaches are proving high enough sensitivity and specificity and to obtain the simplest workflow and lowest costs, which ultimately benefit patients, that maybe multi-omics approaches are not required. Just my thoughts.
Right. Okay, well, that actually brings me to the next question, because that narrows it down to the molecular. And the next question, and as you were speaking there, Chris, I'll ask you this question first of all. So, which technologies for biomarkers do you envision being the most prevalent in the next 5 to 10 years? Is going to be, you think there's going to be one that's going to be better suited to the liquid biopsy than the others? Obviously, I know that for your technology you're using at the moment, you'd hope that to be the best one in the next couple of years. But what are your thoughts on that?
When you say you mean molecular technologies here, Steve?
Yes, I do mean molecular, really specifically molecular, because you mentioned the others being important at the beginning of it, but for the molecular side, what are your thoughts?
So most of the focus for Nonacus is around that MRD space, so kind of post-surgery, post-treatment monitoring, and in that space I do see a continuation of PCR and derivations of PCR due to their simplicity, low cost and sensitivity being an important technology. I also see NGS due to the ability to multiplex and target many loci, which is really critical for certain biomarkers. For example, TMB, MSI, HRD analysis requires very many loci to be targeted at once, and this is out of the range of PCR in an affordable way. So I see these technologies and derivations of them continuing to be prevalent. Maybe increases in the mass spec space, maybe kind of reaching into the kind of MRD space too.
And Shidong, go on, Florent. Go on.
Yeah, and I was going to say again, it's a very wide question, because the biomarkers, which biomarkers will become prevalent, it's down to the research which is currently done also in all the university and research centers. Learning more about the diseases, we will know more about the biomarkers. What we could assume, from an educated guess, is there will be RNA, DNA, or proteins. So that we know for sure. Now, in terms of technology, it's linked to the biomarkers that you want to detect and the specificity of that biomarker. And the good example is the DNA, whether you look at the DNA itself as a mutation, or if you look at the DNA as a carrier of a methylation.
Yeah,
technology will be different. So I think it's hard to predict which one will establish. Of course, qPCR is well established. Even if you look at the current FDA approved tests, I mean, it's probably 3/4 of the tests approved here. So that's a clear indication. Now, you need also to think about the patients, the patient journey. And I think, Chris, you were mentioning the comforts. And, these are drivers for new technology, because we're going to try to answer the patient needs. Whether it is time to results, you want a quick test, quick results, or whether you want something, more suitable for the patient, which could be cost, by the way.
Of course, yes, and that's obviously part of the journey that you actually take. The costing out of what is actually appropriate for the patients themselves, because obviously each technology comes with its own cost. How about Shidong? Do you have anything to say here?
Yeah, definitely. I think that the biomarker will depend on the medicine, and also that I bet there will be a lot more new biomarker coming up in addition to for example, MSI, TMB, PD-L1, and even the targeted therapy marker. But also that the technology-wise, I agree that it will be multi-technology, not only the genomic the with the DNA, RNA, and also that at the proteomic level for protein, but also that the different approach and to address the same question. For example, the flow cytometry and immunohistochemistry, digital pathology, and the AI and machine learning will be all the leverage, and then to form the answer, and then maybe it's a single marker or it's a combination of the different marker. So for the next 5 to 10 years, it will be a package, and there will be a lot more to expect.
Yes, yeah, I totally agree. And I remember a few years ago, because I used to work with microarrays, and the microarrays became the big thing. And then suddenly we discovered there were 500,000 microarrays, and people started saying, well, that's great, but what does it actually mean? And then you had to start asking very specific questions to be able to get the answers that you actually needed. And even then, there was a problem there, because you could actually get the right answer if you asked the right question with the right analysis platform. And that's quite frightening, because if you didn't get the right answer with the right platform, you'd go to another analysis platform, and then another one, until you got the answer that you actually wanted. And this, I think, the technology itself is going really well, but there is a need to make sure that as we carry on developing, that we keep a clear line of sight with what is actually happening with the actual diseases themselves. It's, I think, particularly with the cloud coming along and being able to do a lot more analysis a lot faster is certainly helping. But I know you guys, you probably have your own sets of computer analysis packages to go with your particular assays. Do you think that's a useful thing? Obviously, you've developed your own, but do you think that there is also danger there that you're going to start producing these assays that, because at the moment we're picking the low fruits, we're picking those diseases that only have 1, 2, or a few markers for them. But clearly there's a lot of those diseases out there, particularly cancers, that can have 50, 60, 100 different markers. Some of those are clearly just as important, for the cancer itself. Is there a danger there, do you think?
Steve, I think with the advent of modern therapies like immunotherapies and modern day things like PARP inhibitors, now more than ever there is a need to do complex analysis of liquid biopsies to understand the biomarkers like TMB and MSI, which will make the patients eligible for these amazing new treatments.
Yes, yeah. And we're starting to see that already. Yeah, sorry, go on, Florent.
I like also the Shidong comment on AI, actually, because I can see, with that multi-omic approach for liquid biopsy analysis, the profile and the correlation between every type of data points would be important. And I can see machine learning playing a key role here in probably 5 to 10 plus beyond that, because then you could compile data from PCR, NGS, and then add a layer of methylation, and you have a full individualized analysis. So that could be something also that we see the computational power behind the molecular technology.
So the next question is, how do you see liquid biopsy being adopted for cancer point-of-care settings or oncology specialists? And Shidong, let's start with you with this one.
So be happy to. So liquid biopsy, obviously, that's the best, the material and resources for early cancer detection, because for early cancer detection, you do not know whether a person or individual have cancer or not. And then there is no way that you can take the tissue for any biopsy like a pathology test. And then the liquid biopsy from the body fluid, and clearly just by logic, and it's the right choice. And then this also in complement with the use of the imaging such as the low dose CT in the case of the lung cancer, and like the blood-based or stool or urine or CSF, saliva, and based on the molecular test, they can non-invasively provide the signal towards the early sign of any cancer. So I see that this is the path forward, very clear, and for liquid biopsy to playing a bigger role, not only to diagnose and treat the treatment monitor and also the MRD testing for AIDS patients, but also that for the potential, the people at risk for cancer, or the people already have cancer, but they just never know. So liquid biopsy absolutely is the future, the best choice, and to complement the use of the current imaging and also some of the chemical routine, the clinical testing.
Interesting.
Yeah, and I agree. All the speakers very good opinion, and I think the Shidong gave very overall opinion about the liquid biopsy, and also have a very comprehensive from the different therapeutic requirements, and you have the different way to have the accompanying diagnostic and the biomarker study to support the drug development and the innovation. So that's liquid biopsy definitely play a very important role here. And in the next 5 to 10 years, it will be the major specimen resource there. And I think we need to continue to working on that area to make this. One is from the regulatory viewpoint, how can we make this liquid biopsy become standard? And that's another challenge we also should working on. And the other is how to make that is more accessible for the customer, whatever from the medical side or from the pharmaceutical side, that everyone can access that liquid biopsy methodology and for the diagnostics for the cancer treatment. Thank you.
I'm wondering if there's not an element of answer in the last 2 years we had with COVID. I think if you think about the situation before, after COVID, the population suddenly got educated or acquainted with testing, self-testing, whether it's molecular, whether it's immunoassays. And the facility, the proximity of the testing and all the test centers made that very, I would say, turning nearly into a habit. So if you want to maybe extrapolate a little bit in the future, maybe we could see a situation where we use the same approach. You would follow and monitor your cancer treatments through your own self-testing, or you would have the same services to you post your samples. It will become, I think, it will become easier.
Florent, I think that's a very good opinion around the COVID, making everyone acclimatized to this rapid ready testing and send away testing. But with that in mind, does that lead to point of care testing or centralized lab testing where people post off the results and it goes to a centralized lab? What do you think?
I would say technology, Chris. If your question is complex and you want to have hundreds of lookis, then maybe NGS is probably the best approach. And I see that as a centralized lab. Now, if it's just a yes, no, if there's no quantitative information, then maybe a near patient POC would work.
Thanks, Florent.
Yes, it is interesting, and I think in the next probably 10 years, it's going to become more important, because over the last 2 years, we've had all of those people that should have gone for regular screening that haven't done that regular screening, and there's still a backlog of lots of people that haven't been screened. So we've got that window of probably 3 years now where people haven't been tested, and of course the earlier you can test, the better the prognosis for the patient. So we're going to have some of those patients coming through, and it's going to be important that they get screened as quickly as possible. So the idea of point of care, but not just that, it's also those that have gone beyond that point where they've got the metastases, they've got the spread of the disease, and then you need to then start testing for the additional cancers or that minimum residual disease actually coming up. And the next question is, so what are the unique challenges faced in developing companion diagnostics? I know it's a very, very big question. And as you've been on mute, Harry, let's start with you with that question.
Thank you, Steve. Yeah, I think this is a big question, and for the companion diagnostic, and I think that you need to, from the different perspective, to look at this issue. And one is from the detection side, it's always use the liquid biopsy. Always there's a challenge for the sensitivity and the specificity. So that's a general challenge, always be there. And also from the from the medical society community and the pharmaceutical society, their requirements are different. So the other challenge is the therapeutic area, pharmaceutical industry development is so quick, and right now, not only for the mutation detection, you also have the protein, you have the methylation, and also right now you have the new cellular therapy, gene therapy, all these different therapy. What kind of a biomarker we should detect it, and that is everything you can from the liquid biopsy to get the information or not. It's a lot of challenging and for the development to work. And so that's I think from the different angle, from the technology itself, from the pharmaceutical industry, drug discovery, new requirements, there are lots of different needs there. So you from the different angle, you can see the different challenges there. So I think that's why I just mentioned that liquid biopsy now it's a new area and need a lot of work to continue for the research and for the innovation. And the other thing is from the regulatory side, we also facing the new challenge, how to standardize that methodology and became more compatible, and sometimes even you need the whole package for the testing and detection. So from every angle, you can see that challenge is tremendous.
Absolutely. Okay, Shidong, would you like to make any comments?
Yes, actually, the companion diagnostic is exactly the thing, at Predicine we are doing, and also during my early time at Genentech, and that for drug development program we are doing. So the unique challenge that to develop companion diagnostic, it's really that depending on the program, and the more broadly for any of the medicine, and people want to develop it, to benefit the patient across the world. How you make a harmonized companion diagnostic assay available in US, in Europe, in China, in Japan, and the rest of the countries. And this has been a practical challenge that in front of all pharma and also diagnostic company. And then second is that how you make the assay is really cost the effective and make it affordable. And third is how to you make it accessible. That make the people from the every country in Africa or in India or in China or in South America, they can benefit the same companion diagnostic as the patient in the United States. So I think that basically make the harmonized assay, make it affordable, make it accessible. That's the unique challenge that related to the CDx, the companion diagnostic, and which is the pre-requirement before people can know the diagnostic and take the personalized the therapy and take the right medicine.
I agree with you. I think that 20 years ago I proposed 3A diagnostics: accessible, affordable, accurate. And you mentioned that's 2 points: affordable and accessible. That's very important, and plus the accurate. So that's kind of right now that liquid biopsy add another division and makes the sample accessibility is much better. So that's the plus to realize these 3A diagnostics.
And Harry, I completely agree with your points and Shidong's points, but surely the pharmaceutical industry have a part to play here, because if we look at the current ecosystem around companion diagnostics, it's a pharmaceutical company develop a blockbuster drug, and they want a companion diagnostic which is just targeting the relevant mutations or biomarkers for their drug. But what's best for the patient is maybe a more complex and still low cost companion diagnostic, which actually tells them of all the drugs available that are approved, or indeed clinical trials that are available, which is the best for them. And it's not until the pharmaceutical industry come together and somehow create some kind of funding assistance for this kind of test that it will truly be adopted globally.
Yep, absolutely. Yes, I agree with.
And I think we have to be careful here as well, because we're getting a lot of testing done in the US. I know there's a lot of testing done in China at the moment, but we do have the ethnic stratification. I mean, it's got to be able to be worked in Shidong, you mentioned those other countries. So it can't be just for the US and just for China. It has to work for all of those different countries. Otherwise, you are stratifying the companion diagnostics. Right. Does anybody want to add anything else to that?
For me, the cost is key, because at the end of the day, the patient will have to pay for it. So costs come to the patient, costs come to the developer also. I'm sure Harry, Shidong, Chris, you're well acquainted to the cost of those development efforts. And this is what, for example, we at Meridian have understood, and we try to address and help those guys. Of course, we don't do the test, but we try to provide solutions to whether accelerate the commercialization and develop the development costs. Also make, Harry, your third A, the accuracy of the test with strong chemistry. So I think from what I'm hearing here is really the alignments of the messages, whether we talk about the developers or the reagents or the patients, costs at the end, and accuracy are keys.
Okay, well, we're almost on the 50 minutes now. So what I'll do is, first of all, I'll thank the panelists. That's been great discussion. I've really enjoyed listening to you talking, particularly you, Harry, and now you've got your voice back. But the rest of you, it has been really, really good. If anybody does have questions, please send them to me. You can see my information on the screen there. I'm very happy to answer them, or if you want specific answers, or you want to be in touch with one of the panelists, please feel free to go through me, and I can forward you to the people on here. So really, it just leaves me to say, first of all, thank you very much, Chris. Thank you very much, Harry. Thank you very much, Shidong, and thank you very much, Florent. I think this has been really, really useful, not just for those people listening, but for me as well. I've learned a lot from this, and the technology itself is still in its infancy. We know that. There's a lot to do, and there's a lot more we can do. And the exciting thing at the moment is those technologies are increasing. Particularly, we talked about the analysis. Being able to do more and more analysis faster and faster, being able to get those results to those customers, getting more of the companion diagnostics out there to fit those patients that actually need it. And it's becoming more important for things like prostate cancer, where you've got one cancer, it starts, you give them the drug for it, and then another one takes over, and then another one takes over, and to be able to follow that through, and the minimum residual disease, and actually checking for that. And a lot of this is actually often missed at the moment with just the normal tissue biopsies. So I think in the next few years, we're going to be able to start really getting on top of and beating these cancers. So thank you very much, gentlemen, for your time, and thank you very much for the audience for listening to us.